The other day I recorded one, but forgot to hit the record button. So that was fun. All right. All right. So DNA and RNA structure. DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose, RNA has ribose. And then your nucleotides are thymine, cytosine, guanine, adenine for DNA. But for RNA, we're going to have uracil instead of thymine. So if you ever get a question and it says what kind of nucleic acid is it, and it has thymine in it, it's DNA. If there's uracil in it, it's RNA. And then if they give you like numbers, if your A's and your T's are equal, it's double-stranded. If your G's and your C's are equal, it's double-stranded. But if your A's and your T's are not equal, it's single-stranded DNA. Like if your A's and your U's are equal, then it's double-stranded RNA. So stuff to keep in mind. The additional thing that this diagram does not show is that in our backbone, you've got those phosphates. So remember that you have your sugar, and then you have your phosphate, your sugar, your phosphate, and then coming off your sugar will be your nitrogenous bases. But I don't know how specific that's going to be.
Mr. Irwin, can you repeat the like single-stranded, double-stranded stuff with like?
Yeah, so if you were given a problem and it said you have 20% adenine, 20% thymine, and then 30% guanine, 30% cytosine. So since your A's and your T's are equal and your G's and your C's are equal, you know that it has to be double-stranded so that because they're matching each other. And you know that this is DNA because there's T. But if you were given a problem and it said you've got 20% adenine, 25% thymine, and then whatever G, whatever C, because your A's and your T's are not equal, that would be a single strand of DNA because they're not matching up. And then if you change any of that to U, then it's RNA.
Got it.
Why is the phosphate significant other than stabilizing the sides of the DNA and the RNA in terms of DNA technology? In terms of gel electrophoresis, the phosphates are negatively charged. That's what allows the electricity to drag your DNA. It does. So you could do RNA analysis and gel electrophoresis, and it would also move just like DNA. Okay, why is DNA more stable than RNA? It's double-stranded, exactly. All right. So your basic things. So purines and pyrimidines, remember that those are your A's and your G's. So those are your two rings together. And then your C's and your T's are single rings. And why do we have to have an A to a T and a G to a C?
Apples, trees.
Yes, but why do the cars have to go in the garage and why do the apples have to be on the tree?
Like when they're bigger.
Well, if an apple and a tree go together, how many rings do you have? 3. And if a G and a C go together, you have 3. So that gives you consistent width or diameter with your DNA. Because if you put a G and an A together, it'd be too wide, an A and a C together, it'd be too narrow. And then G's have to go to C's, A's have to go to T's because of the way that they bond. A G and a T would repel each other, an A and a C would repel each other. And that's going to be important for stabilizing the DNA.
Why do you have single probably?
Because you can have a double-stranded RNA in viruses. And you can also have, and also tRNA, there is some double stranding in its structure. And then you can have some gene expression regulation with microRNAs and and other RNAs as well. Okay, good. Yes. All right. DNA is semi-conservative. So this is what's meant by conservative. The original separates, you get the copy, the original comes together. And then dispersive, it just randomly breaks apart. And so all your DNA is broken in bits and pieces of new and old. Semi-conservative means they separate and then each side acts as a template for the other. Semi-conservative is the winner. Probably this will not be asked, but this was the Mendelssohn-Stahl. Experiment where they used the heavy nitrogen to figure this out. If that comes up, they will give that information to you, and then your answer would be, if you see Mendelssohn and Stahl, somehow the answer is going to be about semi-conservative replication. So they separate, this acts as a template, this acts as a template, and then you get new DNA built by those. And everyone together, DNA is built in the, okay, let's do that again. Everyone together, it's built in the, it's really sad, 5' to 3' direction, because I'm sure the recording did not pick that up. All right, so when we are talking about DNA replication and all of the enzymes, we are looking at, a little frustrated back there. So our first one, we've got the topoisomerase, and the job of the topoisomerase is to kind of undo the supercoiling of the DNA. Why is DNA supercoiled?
Storage.
Right. You've got 6 feet of DNA in every nucleus. That's got to get packed down. So that's your DNA packing. And so we have to unwind that so that we can replicate it. And so after we undo the supercoiling, then the helicase is going to separate the strands. And when we say it's separating the strand, what is it breaking to separate them?
Hydrogen bonds.
Correct. How many hydrogen bonds between a guanine and the cytosine? 3. How many between an adenine and a thymine? 2. And so guanine cytosine bonds are stronger. So if you have a DNA strand with more guanine cytosine and another strand that has more adenine thymine, that's going to make it a stronger DNA molecule. All right, so DNA, once it separates, it wants to go right back. That's called annealing or reannealing. And so we've got our single strand binding proteins to prevent that from happening. So at this point, we can start building DNA, correct?
It prevents them from
Going back together or reannealing. Once we've opened up the DNA, we can just start building DNA. No, what do we need?
Primates.
We need primase to build our RNA primer, because we need a foundation for our DNA polymerase. And then our DNA polymerase is going to start building off of that. One strand will be built continuously. That is the
Leading strand.
all right. And the other one will be built in fragments.
Lagging.
Lagging strand, and the fragments are called
Okazaki fragment.
correct. And in order to bind the Okazaki fragments together, we need ligase. So a question that they like to ask is, a scientist was doing an experiment, blah, 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 and found that there were many fragments of DNA after the process of replication. Which enzyme is probably faulty? And the answer is ligase, because it's not binding the Okazakis together. Yes.
What replaces RNA primates in PCR?
So there is another DNA polymerase, DNA polymerase 1, that is going to replace it.
Why did you say the RNA primase is essential?
Why is RNA primase essential? That builds the RNA primer. And that's your Lego base plate to build your stable Lego tower. All right, so the DNA polymerase that actually builds it is DNA polymerase 3. DNA polymerase 1 removes the primer and then replaces it with DNA. And then after that, your ligase will bind all of your Okazaki fragments together. Good. Give me a second.
Tac polymerase is in PCR, right?
Tac polymerase is in PCR. Why do we use Tac polymerase? It is heat resistant because remember it comes from bacteria from the hot springs, and that keeps, so it's able to handle the heating and the cooling of the thermal cycler in the PCR. And then what do we replace the RNA primers with in PCR? So we have RNA primers. Why is this essential? What replaces the primers? Tac polymerase doesn't replace the primers. So what replaces the primers, instead of RNA, we use DNA primers that we add to the mix. So you're going to have a forward and a reverse primer so that we don't have Okazaki fragments.
Wait, I'm confused, because when I asked earlier, like what replaces the primase in PCR, you said DNA polymerase 1. Is that?
Oh, I thought you were talking about what actually replaces the primers in the, no. So what replaces primase in PCR are the DNA, the artificially built DNA primers. So like, you know, when like downstairs when they're doing DNA sequencing and they're doing, you know, their PCRs, they have to figure out what primers to do. And so those are artificially built primers, and just add them in the mix. Okay, Okazaki fragments, why do they form? Sorry, what?
Lagging strand. That's
So-
the other way.
All right, so DNA itself is anti-parallel, meaning, right, the strands go in the opposite direction. One goes 5 to 3, the other one goes 3 to 5. And so polymerase can only build by prime to 3 prime. And so this has to wait. So in order to build 5 prime to 3 prime, your DNA has to open enough to start building. And so then as this opens up more, our leading strand just keeps building this way. But then when this opens up enough, then we'll build another segment going that way. And so it's the fact that DNA is anti-parallel that you have to build one side in short segments as the helicase unwinds the DNA. We avoid that problem in PCR by having forward and reverse primers, so you can go both directions as leading strands. Because we don't use helicase to unwind it, we use heat to separate the molecules, so we can build the new DNA. Central dogma, DNA to RNA to protein. This is the most important concept of biology. So DNA to RNA is transcription, and then RNA to protein is translation. And so all of this, we talked about DNA as being the control center of the cell. Yes, because it makes the RNA. It's the RNA that does all the work. All DNA does is kind of like lay there and get copied. But RNA does the actual work of building the proteins to make the cell run. So we've got lots of different kinds of RNA, which have different shapes. Yes.
The question says like explain in terms of code, codon, and anti-codon.
Excellent question. So DNA is our code that is copied into the RNA. And so every 3 letters of the RNA is the codon. And what does the codon, what matches to the codon? And what is the anticodon? It's the tRNA. And so then those 2 work together because remember that our tRNA brings in our amino acids. And so these 2 work together to do the translation part. So these are the instructions right here. The copy of the instructions is the codon. And then the actual translator is the anticodon because that brings in the amino acid information, matching it to the nucleic acid information, converting one molecular language to the other. Okay, so mRNA, linear, so big long ribbon, and then that's our codon sequence for protein synthesis. Our tRNAs are going to be folded. So we've got some double stranding in there. That's going to be the anticodon. That's going to bring in the amino acids. Then we have the rRNA that is going to form the ribosomes or the ribozyme. Why are we using the word ribozyme? Correct. And that is also some of the evidence for the what theory? Not the endosymbiotic. Before that,
RNA world.
the RNA world hypothesis, right? Because it is acting as both an enzyme and as a molecular inheritance molecule that can self-replicate and do the job of proteins. MicroRNAs and small interfering RNAs. This is post-transcriptional gene regulation. So what do we mean by post-transcriptional? Not the post-transcriptional, not post-trans. So after the mRNA has been made, this shuts down the mRNA, so it stops making the protein. Pre-transcriptional regulation would be things like the operon systems and the enhancer and silencing sequences. snRNAs are the ones that make the SNRPs that do the splicing of the introns and the exons. And then the signal recognition particle, that's what SRP stands for. This is what tells a protein that it's going to go into your endomembrane system, which is your ER to vesicle to Golgi to vesicle to secretion. So when your immune cells are making antibodies, those proteins, when that protein starts to get made by the ribosome, an SRP will attach to it, tell it to go into the ER, and then it'll get shipped out. If it does not have an SRP RNA attached to it, that protein is going to stay in the cytoplasm. Good, almost. Okay, so RNA structure, so we've got, so this is both question 7 and 12, which will hit 12 again, but you've got your primary transcripts. So that is your pre-mRNA. To your pre-mRNA, you're going to add your GTP cap or your 5' cap. And then you're also going to add your poly A tail. Lots of adenines, like 200 to 250. You also have your introns and your exons. The introns will be removed. The exons will be spliced together. This is done by the signal, the SNRPs, the small nuclear ribonucleoproteins. But let's just back up. What's an intron and an exon?
Introns, the parts that don't have useful information, right?
So these are not expressed. And these are expressed. And so those introns have to get cut out. And then the exons spliced together. If the introns remain inside of the mRNA, your protein will not be formed properly. And then you will, and so again, that's part of the problem with putting eukaryotic genes into prokaryotes, because prokaryotes do not do this process. And therefore, we have to make copies of the RNA without the introns, which is called cDNA or complementary DNA. tRNA structure, the important things that you need to know is at the 3' end, that's where we have the amino acids attaching. And then at the other end, which is not the 5' end, because the 5' end, because of folding, the other end, this is the anticodon, which is going to match to our codon down here in the mRNA. And when they are binding, remember that this third nucleotide match isn't always super exact. So that can lead to the wobble effect. I know that I'm writing that down there and you all can't see it. But what does the wobble effect do?
So like what's all the amino acids be built from only? Like,
So
I'm
they
thinking
don't
back
fit,
now.
so then it's like,
So then it causes a redundancy in how we interpret the thing. So you've got maybe 4 different codon sequences that bring in the same amino acid. And so if you have a mutation on that third codon, it may still bring in the same amino acid. And what kind of mutation is that called?
silent.
Correct, it is a silent mutation. All right, ribosome structure. So we've got the large subunit, we've got the small subunit, and then in the large subunit, you've got the E site, the P site, and the A site. The small subunit's job is simply just kind of sandwich the mRNA between the two, kind of it's a holder, like a binder clip. And then, of course, remember that you have your building protein in the P site, your new amino acid, your new tRNA comes into the A site, your anticodon matching to your codon, and then we'll transfer those, everything will shift, things go out the E site. I mean, specifics on how they would ask stuff about that. I've never really seen specifics on that in the multiple choice. It's more about interpretation and using the codon sequence to build proteins. MR, the microRNAs and the small interfering RNAs, what they do is they bind to messenger RNA. And now my ribosome can't move past that. And so that's why it's post-transcriptional gene regulation. So that prevents the protein from being made. So we see that here. And so when this happens, translation gets blocked and then your RNA degrades. And so that gene no longer is getting expressed in that cell. snRNAs, these are the ones that make the SNRPs. So cutting out the introns, binding the exons together for gene or for mRNA processing. And then your SRP RNA, that's the signal recognition particle, binding to the protein coming out that directs it down to your rough ER. And then eventually this protein will get secreted from the cell. So that gives you two kinds of ribosomes, cytoplasmic ribosomes and bound ribosomes. Bound ribosomes are only bound if the protein they are making has this sequence on it. So there's actually no difference between the ribosomes bound, and it's just the protein that they're making determines where they go. All right, so looking at this, we've got lots of labels. We're going to label all of them, and I'm going to tell you which ones are probably the most important. So going from DNA to RNA is what? That's going to be important. RNA to protein, also important. And then we've got all these other terms labeling the DNA. So then let's do these. So then the code is your DNA. And this is your this is your codon. And then this, and then your tRNA down here. That's your anticodon. Okay, so our RNA has been made. Our RNA is going 5' to 3'. Did it copy the blue strand or the yellow strand of the DNA? The yellow, because the RNA is built 5 to 3, so it has to copy the 3 to 5. So when we talk about that, the yellow strand is going to be your template, which means that the blue strand is the non-template. So that means this is what's going to be known as the sense strand, probably not that important to know. And this is the nonsense strand. And so this becomes, so this is what's called the code. And then this is called, actually, let me cross these out, I'll confuse myself. And so this is called the coding strand. And then this is the anti-coding strand. The language that they will probably use on the AP test is the template and then maybe the code. Actually, switch those. They'll use template and code. So this is the code. So the code is actually going to match it's going to match this right here, because this is the, well, it's going to match the opposite because this is the anticodon, which matches that. So
Wait, so the blue and yellow is the coding strand.
So this is the template strand, and the other one is called the coding strand, which doesn't make sense because, like, wouldn't you copy the coding strand? No, you copy the template strand. Like, why is science so bad at naming things? on the AP test, so again, this is stuff I emphasized over and over again. If they give you all of these strands of DNA and RNA, look at your RNA, identify the 5' end, and then know that it has to copy the DNA strand that is 3 to 5 opposite. Yes. So yellow, no, leading and lagging is for replication. This is for transcription. So if I can find them in my list of practice tests, there's a specific question that I want you to do on the quiz that is all about like which strand goes with which, but just look for the 3 to 5 that makes the 5 to 3 to read it. But when we're looking at this will be on there probably in some form or another. This your codon chart. So remember, it's the codon chart. So right here, this is the tRNA, that's the anticodon. That's what brings in the amino acid. That's not what we want to look at. What we want to look at is the codon sequence of the RNA, of the mRNA. So then AUG is going to bring in start methionine. So if you can't remember that, it's going to be on there, but there are probably 4 codons that you need to know. AUG is start, and what are the other 3? Your stops. UGA, UAA, and UAG. If this chart is on there, it's going to tell you that because stop is UGA, and then UAA and UAG are right there. But if it's not on there, which I would be surprised, just you need to know those because it could ask you questions about nonsense mutations and things like that. So if that is start, what would GGG give you?
Bottom right corner.
So GLY, so that'd be glycine. CUC,
Says it underneath.
and then AGC, serine, et cetera, on and on. So a lot of time they may give you a sequence of DNA and this chart, and they will ask you what the amino acid sequence is. If they give you DNA, you have to transcribe it. They may give you an anticodon sequence. And they give you the anticodon sequence, then you have to flip it. So you have to make sure you know what you're looking at, and you have to interpret it from the mRNA. Yes.
Practice problems like that.
Yes. In here,
Or are they only the other?
I don't, but I will have them on your quiz. So, yes, yes.
So like template strands makes,
The template strand makes the mRNA. That's the important one.
so that's the codon.
So the code, so that's what, so you have the coding, you have the code, which is the non-template strand. So that is opposite the template strand.
I'm
So-
very confused by these questions, 9 and 10.
Oh, 9 and 10. So how would you use the codon chart to determine the amino acid sequence? You would just do the thing with the box. The question above that, using above diagram, if you were given the non-template strand, which would be the blue, would that be equivalent to the codon or the anticodon? So ATG gives you what in terms of RNA?
UAC.
So that would be equivalent to the anticodon. All right, transcription. you're going to have your promoter sequence. In your promoter sequence, you will have the TATA box, the sequence of thymines and adenines. The importance of the TATA box is where the transcription factors bind. And why do we need transcription factors? What do they guide in?
The mRNA.
Well, we're gonna make mRNA.
Yeah.
This is the DNA. So it guides in RNA polymerase. To guide in the transcription factors, you have these upstream enhancer sequences. So this is pre-transcription control. All of this goes back to signal transduction pathway. So the hormone comes in, the hormone could potentially be transcription factors. The hormone could be activators that bind to the enhancer region. And so then they start off the process of transcription. And then what is the RNA polymerase going to do? It's going to do two things.
Bill
The RNA.
separates.
Separates the, unzips the DNA and then makes, this is transcription, this is not replication. So RNA polymerase does the job of helicase and DNA polymerase, but instead of building DNA, we are building RNA, so that's transcription. And so then our transcription direction is we are building 5 to 3. So we're reading our DNA 3 to 5. And so again, using other terminology, the sense strand, which is also the coding strand, which is also the non-template strand. So I don't know why we have to have 16 different words for the same thing. Yes.
So if you're making an RNA, like, yeah, if you're like making an RNA strand, you take the DNA 5'3". You essentially just like put it backwards and then like match the like A's.
No.
Okay, what?
So,
I'm so confused.
so
I can read it 3 to 5 then.
let's turn this to the board. So if we have a DNA strand and my DNA is running 5 to 3 this way, my opposite strand is 3 to 5 like that, anti-parallel. So let's say that this is where my promoter region is on that end. So as the polymerase opens my DNA, it is going to be building my RNA 5 to 3 that way. So it is copying this bottom strand. So because we are building 5 to 3, we have to read the DNA 3 to 5.
So you're not reading.
We're
the
not
same-
reading this,
sorry. Okay,
we're not reading this strand.
okay. I
Unless
thought we were reading this.
the promoter region was down here. And then we would build 5 to 3 this way. So it all depends on where the promoter is in relation to. So the DNA is always going to read, DNA is always read 3 to 5, whether you are replicating it or you are transcribing it, because we have to read 3 to 5 to build anti-parallel 5 to 3. All right, RNA modification. We've already done that. So we cut out the introns, we put on 5' cap, we put on poly A tail. As we are taking and modifying RNA, we can also do alternative RNA splicing. And so what alternative RNA splicing is, this is our gene right here, this whole thing, this is one sequence of DNA that is copied into the RNA, so transcription. And we've got lots of different exons. So we cut out all the introns every time, but we can also cut out exons. And if we leave all of them in, these will all get translated into this protein. But if we cut out the third exon as well as some introns, our sequence of what's being read will now be different. And so then we get a different protein, very similar, but slightly different. And then if we cut out a different exon, we get a third protein. So what this tells us is that we can sometimes get multiple proteins from the same gene, depending on alternative RNA splicing. Now, once we are to the point of translation, so again, a quick overview, we are going to have our small subunit bind to the beginning, and then the first tRNA, which is going to match to our AUG codon, that is going to bring in start methionine. The large subunit comes in so that is in the P site to start. This is our A site, our E site. The next tRNA comes in with the next amino acid. We transfer the amino acid or the growing protein to the top of that. That is done by what? What does the transfer? The large subunit. So that's catalyzing a reaction. That is dehydration synthesis when those are bonding. That's an enzymatic reaction. So that's why it's the ribozyme. And then everything shifts until we get to a stop code, and that keeps going and going and going until we get to a stop codon. And then you get the release factor. And so when this goes to shift to the release factor, there's no amino acid. So there's no bond, and then that separates and everything dissociates. It is extremely, extremely unlikely that you will have to explain the process of translation. But you should be familiar enough with it that they said, you know, I don't know, who knows what they'll even ask. It's a mystery. So, all right. So another picture of it. Cytoplasmic versus bound ribosomes. So bound ribosomes are attached to the ER. Non-bound or cytoplasmic are the ones floating in the cytoplasm. What's important, these free ribosomes, these make cytosolic enzymes and proteins. So things like all of the enzymes that need to run glycolysis, like phosphofructokinase. The bound ribosomes, again, these make the proteins that are either going to be secreted from the cell or going to be attached to the cell membrane. And that's regulated by whether or not there is this signal peptide, which binds to your signal recognition particle. And so that's all about kind of post-transcriptional and translational control of where those proteins end up. So if there's a mutation in your DNA sequence, which alters the structure of this signal peptide, so it's not there, that protein that's made is going to end up where? Floating around in the cytoplasm. And if this protein, maybe that's going to become part of the cell membrane, and if that's an important signal recognition particle protein, or if that's a transmembranal protein, then that function of that cell is going to be gone. So, all right, protein synthesis in prokaryotes versus eukaryotes. There is no nucleus in the prokaryotes. So this is my prokaryote. So there's no nucleus. The defining definition of a prokaryote, which sometimes shows up on the AP test. And I never want to know if anyone gets that wrong. But what happens, what is significant is transcription and translation. They can happen at the same time. So as soon as transcription starts, a ribosome can bind and start making the protein even before transcription is done. Because prokaryotes, there is no RNA modification. In the eukaryote, because we have a nucleus, transcription happens in the nucleus, RNA modification happens in the nucleus, and then translation happens in the cytoplasm. So, but you still can have polyribosomes in both cases. What do we mean by polyribosomes?
Them working on the same strand.
Right, so lots of ribosomes reading along the mRNA, making lots of proteins all at the same time, making it more efficient. All right, DNA, important?
Yeah.
Yes, DNA is a sign of a common ancestor of LUCA, the last universal common ancestor. And the main reason is all organisms have it. And so it's called a molecular fossil. And the reason that it's also other evidence is we can take eukaryotic DNA, see ya, and we can put it into a plasmid and put that into bacteria, and the bacteria can read any DNA. Any cell can read and transcribe and translate any DNA. It is a universal code in every type of cell. So that is an indicator that the mechanisms of transcription, translation, gene expression all came from some common ancestor, which they call LUCA. All right, viruses. Our basic viral infection patterns are the lytic cycle, the lysogenic cycle. Both of these are for bacteriophages. So lytic, what's the big difference? Lytic causes what causes the cells to break open, whereas the lysogenic cycle is going to encode the phage DNA into the bacterial DNA. And so that's called a prophage right here where it's incorporated. And so it just stays in there, reproducing as the bacteria reproduce. So this doesn't kill the bacteria. The bacteria reproduce until there is some environmental change, and that shifts it back into the lytic stage so that you can do massive reproduction of the bacteriophages to spread to other bacteria to hopefully get out of that bad environmental condition. So this is just for bacteriophages and bacteria. Significant things that might show up, they might mention the Hershey Chase experiment. What did the Hershey Chase experiment show? Using the radioactive phosphorus, the radioactive sulfur, bacteriophages and the bacteria, it proved what?
Was it like DNA?
Right.
Yeah.
It proved that DNA is the molecule of inheritance instead of proteins. Why did they think proteins were the molecule of inheritance before that? Right, 20 amino acids versus 4 nucleotides makes more sense that you get more traits out of 20 than 4. The other viral infection pattern is your enveloped viruses. So things like flu and COVID, they've got this envelope around them. The spike proteins trick your signal transduction pathways to draw the virus in, and the virus disassembles, takes over the host cell function, and it makes new viruses. So looking at this pattern, what we see are little spike proteins, which we heard so much about with COVID. They bind to the surface cell, that brings it in, the virus, then your DNA or RNA, depending on what kind of virus it is, makes all the parts. And so then this is also important, these new spikes, they get on the surface of the cell. So as the virus exits, those spike proteins are on the surface of that. But why is it important that these spike proteins are now on the surface of this infected cell?
For antibodies?
So antibodies can bind to that, but this can become a antigen presentation cell, and then this will alert your T cells to come kill it. Because this has now become a non-self cell, which gets targeted and killed. But this is also why viruses tend to be species specific, because they bind to these specific cell receptors. It's also why viruses tend to be specific to different tissues, because different cells have different receptors. So if a virus can infect either different species or different tissue types, it's because those cells have common receptors. All right, a retrovirus, what we need to know, this is an RNA virus. When it gets into the cell, it makes an enzyme called reverse transcriptase. And what is reverse transcriptase going to do with the RNA of the virus? Take the RNA, DNA, and then the DNA makes new viruses. Most famous, is COVID reverse transcriptase? I don't know. HIV is a retrovirus that causes that to happen. All right, DNA expression control. So we've got histone proteins. What do these do? What are they responsible for? DNA folding. And so if DNA is super closed up, will it be expressed? What is that, hetero or euchromatin? It is heterochromatin. So heterochromatin is tightly wound. So it's not expressed because your proteins cannot get in there. So you have to have loose strands of DNA. So this is going to be our euchromatin. And so that's going to allow our gene expression machinery to get in there and copy the DNA to RNA. We can take euchromatin into heterochromatin if we methylate it. So methylation is going to condense our DNA, turning it off. But if we've got histone proteins, we can also acetylate them. And acetyl groups, when we add acetyl groups, this unwinds the DNA to allow it to be expressed. Methylation is usually irreversible, not always. And then, so these two things, methylation and acetylation, is what kind of genetic control? That is epigenetics. And so, because this is stuff that is written on top of, so epi, like your epidermis on top, that is on top of the genes. And again, you read that article about the rats and how different environmental things can change the epigenetic factors. Drinking green tea, that can demethylate tumor suppressor genes. So tumor suppressor genes are the genes that suppress tumors. If those get methylated, then your tumor suppressor gene is off, which can then lead to tumors. So green tea have been shown to have some demethylating abilities. Okay, operons. Actually, one last thing about heterochromatin. Bar bodies, mammals are going to have one X chromosome that gets highly methylated into the bar body. It turns completely off. So this is why you can have heterozygote conditions being expressed as if they are separate in a eukaryotic mammal that is female. So that's why you have the brown and the orange fur, because of the bar bodies. You won't have that in males unless it is due to non-disjunction and you get an XXY male. So all right, upstream regulation, we already talked about this. You have this enhancer region activator guide in your transcription factors to help with gene expression. And then you also can have silencers that do the opposite. So these guide transcription factors to promote gene expression. So again, these activators could be made due to a hormone saying, activate, go. All right, prokaryote operon. So remember that an operon, you're going to have, yes.
What is silence?
So silencers would do the opposite. They would block transcription. So operons, you are going to have a regulator gene. Regulator gene makes the repressor. And so then the repressor will bind to the operon at the operator, and the repressor blocks transcription. Now, the question is the operator, or is the repressor on, or is the repressor off? And then that's going to be the difference of how the repressor gets turned on and off. That is the difference between a repressible operon and an inducible operon. And so is this inducible or is this repressible? Because
When the thing goes in, it starts.
so when the thing goes in, it starts. Exactly. So lactose, when it binds to the repressor is turned
Oh,
off. So this is now off. And so you can have transcription. So in bacteria, in the presence of lactose, we want to turn the repressor off so we can make lactase to digest the lactose. And then once all the lactose is digested, this turns back on, binds, and we stop making lactase. The opposite of that is going to be the repressible operon. So when you have lots of tryptophan, it binds to the repressor And now this is on. And so then we stop making the enzymes to make tryptophan. When the cell uses up its tryptophan sources, the repressor is off. And so then we make the enzymes that will then make tryptophan. And when there's enough tryptophan, it will go and turn the system off. What is that? When you have enough product that things shut down.
Negative feedback.
Correct. So the two most common operons that show up on the test, the trip operon, repressible, and then the lac operon is inducible. So how can we remember that? Trip operon. So R for repressible. And the lac operon, lac stands for what sugar?
Inducible.
Lac is lactose. Where do we find lactose? In milk. So I for inducible. It's a stretch. It works. Got it? Good. All right, cell differentiation. Basically, you start out with a totipotent cell that's going to then make your embryo, and then you'll have your placenta, and actually it'll make the placenta and the embryo. Then the embryo cells are pluripotent, which means that they can become any tissue type. And then the tissue types will become more specific and become multipotent. And so then a multipotent cell would be like bone marrow cells. And then they can become any type of blood cell. But these bone marrow cells, they cannot become skin cells or lung cells or whatever. And so what's happening as we go from totipotent to pluripotent to multipotent, this is where we are having histone and methylation, and we start turning off genes permanently. And so once these turn off genes, then that is going to cause those cells to become more specialized. And so then, certain genes that are needed to make skin cells are turned off in the bone marrow cells. Certain cells that are active here are not active over here because they've been turned off by the methylation and the histone folding process. So turned off genes, so this is your heterochromatin. And so what scientists have been able to do is they can take something like a skin cell and then they can reprogram it back into a pluripotent cell, and that's called an induced pluripotent stem cell. So rather than having to use frozen embryos, they can skip the ethical barrier and just make the cells that they need. DNA mutations. So again, we can have a frame shift mutation. We insert an extra letter, so it's usually a replication error, could be a transcription error. And what's this going to do? Why is it called a frame shift mutation? Shifts everything down. And then we can have a silent mutation. The mutation brings in the same amino acid. Any effect? No. And then we can have a missense mutation where it brings in a different amino acid. And that could change the shape of the protein. What is our example that we've talked about over and over and over again? That is a missense mutation, sickle cell anemia. And then a nonsense mutation, we have a premature stop codon. So then your protein gets cut off early.
That's the worst one, right? Talk about,
Yeah, because that protein is just going to be non-functional. So like even with sickle cell anemia, it's still has a little bit of function. It's more negative than it is positive. So then this is frame shift. So this is a deletion. So we cut that one out. So everybody shifts that way. So all of our amino acids are different. And then insertion, it just shifts this way, causing differences. Chromosomal errors. So these are non-disjunctions. So what we see is we either have the homologous chromosomes not separating properly, or we have the sister chromatids not separating properly. And this results in incorrect numbers of chromosomes. And so what's our most known example of non-disjunction? Correct. The answer to that was trisomy 21 for the recording. We can also have lots of errors either from crossover. So during crossover, you can lose some genes, that's deletion, or when you have a deletion, that means the other one is probably getting duplicated. So you would lose information here. Here you would gain. So if you remember that picture, that giant, like massive cow, the Belgian blue, because it had a duplication in the muscle building gene. So it's just giant and stacked. And then inversion is where the genes get flipped. So all those would be read backwards. So the proteins would be made incorrectly. Insertions or translocations, all of these are where basically you have crossover with non-homologous chromosomes. And so then you get lots of, weird information. The difference between vertical and horizontal gene transfer. So with the DNA that you got from your parents down to you, that is called vertical gene transfer. So when you go from, or when bacteria just split in mitosis, that's vertical. Horizontal gene transfer, that would be when you get DNA from one bacterium to another. So transformation, remember that is simply going to be using plasmid transfer. No, that is, sorry, let's flip that. This is plasmid transfer. Transformation is from the environment, and then transduction is when the virus, the bacteriophage, transfers bits. And so if these are helpful genes like antibiotic resistance, then you could have some genetic recombination. And remember that conjugation is when you have the pili growing and they exchange through the little tunnels, stuff like that. So this is like as close as you get with sexual recombination with bacteria through horizontal gene transfer. So transduction, the virus transfers it, dead bacteria, transformation gets picked up from the environment, then conjugation, transferring that plasmid over. All right, quick on DNA tech. This is, why does it work?
Negative charge.
DNA has a negative charge because of the phosphates. The gel is made of agarose. So we put our DNA in our wells. The positive electrode is down here, the negative is here. The electricity draws them, and these are the fragments that we use to analyze the DNA. So if I had another sample that looked like that, we would say that these were similar. So one possibility that you could be given on the test is you get a gel from multiple different species, and then you have to draw a cladogram from that. So the one that is different from all the others would be the one on the far, which is called the out group. Good job, everyone. So let's just make up an example. So we'll do this one. We'll do. So if we call this A, then we'll call that one E. So who would be our out group? A, because it has the least differences, which would be our top two probably.
Or no?
B and C.
That's hard. Well, so let's do this. We have two pairs that would go together. So which two pairs would probably go together?
C, oh wait, no.
B and C and D and E. So which ones are probably closest to A? B and C, because they've got that. So we probably put B and C here, and then we put D and E there. Does it matter that I put D here or E there? No, it doesn't matter. So that's one possibility that you could get from that.
Does it matter that D only has two bands? So does A.
That D has two and A doesn't actually
I
have
mean,
two.
No, A has one and then the rest have three
So this one has four.
or four.
So, I mean, I just made that up on the spot. So it does matter, but I don't know how much my made up example is accurate. So, but the general idea. All right, DNA tech polymerase chain reaction. So we're denaturing. We're adding our forward and our reverse primers. So we can build 5' to 3' both ways, so we don't make what? So we don't make Okazaki fragments, correct? And then we let it cool, it builds, we use Taq polymerase, it's heat resistant, and repeat this about 30 times. We get billions of copies of DNA, focusing on our target sequence. What gives us a target sequence? What's the thing that makes, what targets the sequence that we want? The primers do that. Otherwise, you'd be copying all the DNA, which is too much information. And then bacterial transformation, this is what we did with the glow-in-the-dark bacteria, the DNA lab that actually worked. So we have plasmids that have genes that we want. We heat up the bacteria a little bit, they absorb the plasmids, and then we grow them. And what is almost always going to be incorporated in that plasmid for selection of the correct bacteria? What kind of gene? You could use the glowing one, but antibiotic resistant. Yeah,
You know what I mean.
I do. If you didn't have the antibiotic resistance gene, what would this plate look like? It'd be completely covered if you didn't have antibiotic resistant genes, because all the non-transformed bacteria would also grow. Where is the antibiotic that we're resisting? Yeah, the antibiotic is it's in the agar, And so actually, if you didn't have the antibiotic resistant gene, nothing would grow. So you need that antibiotic and you need the antibiotic resistance gene so that only the things that have been successfully transformed grow. Otherwise, it's a needle in a haystack. All right, other DNA tech, sequencing. So they run it through a machine and it tells you the order. You will not have to explain Sanger sequencing. The only thing is that you will possibly be given DNA sequences to do cladograms or to identify similar species, etc. That was very long. Thank you, people. There will not be a quiz on Monday.
So when are we gonna take on these quizzes?
I'm gonna make kind of like one evolution and DNA quiz. So we'll have that on, let's say Wednesday. So, all right. I'll change